this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
Today, I'd like to welcome to the podcast Timothy Sangster. Tim Sangster is an experienced and well-traveled bioanalytical scientist, having spent time in Scotland, Italy, England, and the USA. He has experience working for both the contract research organizations and pharmaceutical companies, and has recently, as of July 2020, started his next exciting opportunity as the Executive Director of Bioanalytical Sciences for Celeron in Lincoln, Nebraska. Tim's new role is an excellent opportunity to work in a large experienced team offering a complete range of chromatography, ligand binding, molecular biology, and immunological endpoints to support our customers in meeting their research goals. A visible and highly engaged member of the global bioanalytical community for over 20 years, Tim is also proud to be the chair of the International Reed Bioanalytical Forum, which has a focused bioanalytical meeting that happens every 2 years and has been running since 1975. So today I'd like to welcome to the podcast Timothy Sangster. Welcome, Timothy.
Thanks, Don. It's my pleasure to be here.
Very good. Yeah, so we're going to have a great conversation overall about things that are going on overall in hopefully your life and career, but also a little bit about bioanalysis and what that specifically is. I guess where I'd like to start is, could you tell the listeners a little bit about your career and what you're doing now as the Executive Director at Celeron?
Oh, absolutely. So my career started over 25 years ago. I was very lucky to get a job at Quintiles in Edinburgh, working as an analyst in the laboratory and doing, and I'll explain a little bit more in a minute, bioanalysis. From there, I took my career on a little bit of a global trip. Spent 5 years in Edinburgh working for Quintiles. Then I got the absolute amazing joy to go and work for Pharmacia, just outside of Milan in Italy. And I was working over there doing drug development, primarily oncology. Unfortunately, they got bought by Pfizer while I was there. And I kind of made the decision at that stage to make my own move to try and come back to the UK. Then I jumped ship from Italy back to England to work for AstraZeneca. And I had a great opportunity to be a research scientist with AstraZeneca. And at that point, I spent just over 3 years, and I was approached to take an opportunity in the United States to head up my own department working for Huntington Life Sciences just outside of Princeton, New Jersey. It seemed too good an opportunity to pass up. So I jumped across the water to go and work for HLS. Unfortunately, while I was at HLS, I was loving my job, loving my team, had a great bunch of people around me, and we were doing some amazing things. My mum was diagnosed with fibrosis of the lungs. I'd also had three children at that point. And so my wife and I had a chat, and we decided to look for an opportunity back in Europe so that the girls could get a chance to meet their grandmother, and we could be closer to family while my mum was going through, and my dad as well. Then I got a phenomenal opportunity with Charles River to head up a multidisciplinary bioanalytical team covering everything from basic small chemical molecules all the way through to biologics and some of these new modalities that are coming through the processes, cell and gene therapies. Charles River for 10 years. My mum unfortunately passed away, but luckily we were back and able to spend some time with her. And then I took the chance to jump to Eurofins to head up my own site down in England. But before I actually joined and before I started my job, they decided to close the site. So that unfortunately then put me back looking for an opportunity. I was fortunate to be able to take my time, and the opportunity with Celeron came along. And so in July of this year, I joined Celeron, which is a primarily clinical business with clinics in Phoenix, Arizona, Lincoln, Nebraska, and Belfast, Northern Ireland. I'm the executive director for Celeron's bioanalytical services based out of the Lincoln site. Although at this moment in time, I'm sitting in Scotland still due to the global pandemic. And my role is to lead a team of over 100 scientists who support bioanalysis. And the simplest way to describe bioanalysis is when we talk in the public domain about athletes being tested for drugs and being found that they've been taking the wrong drug, or they've been taking drugs they shouldn't have been taking, particularly around steroids and amphetamines, etc. The tests that are used in sport is very similar to what my team do. We test blood or any other biologic product for the presence of drugs, or in some cases for things that should be there, because we're looking for changes in biologic activity. So we're looking for increases in something in the body that's normally there, which we call a biomarker, or decreases of it. And so my team developed the assays for that. They test them, they qualify them, or validate them, as we call them. And then we run the analysis and generate the data back. And our role is not for drugs in sport, but our role is for the pharmaceutical business. So we do it testing those analyses to help support drug development. Do you have any questions on that particularly?
No, it's very good. So yeah, I think it's really interesting to see how the science aligns to provide more details in support of what it is that you're actually seeing and looking at. And yeah, bioanalysis is definitely an interesting field, and congratulations on your new role. And yeah, hopefully you'll get to come across to the U.S. here soon. Hopefully this pandemic, we find our way to work with it. I don't know necessarily that it's going to go away, but we will probably have to find a way to work and live in an environment with it.
I'm sure we will. If you think back, we've had so many different diseases and things that we live with all the time. It was really interesting that, there was a published study from the Imperial College in London showing that after exposure to COVID-19, people's antibodies drop off rapidly. And so the question is, does that mean we'll never be? The immune system's incredibly complicated. And if you think there's quite a lot of vaccines out there, and it was my wife that pointed out to me that if you look at the tetanus vaccine, you actually have to have 3 shots. You have to have 3 exposures before you generate full immunity. So it could well be that COVID-19 becomes something like that. We'll find a way around it, human beings, and we're incredibly clever and innovative, and we'll come up with answers and come up with solutions. But it's just unfortunately going to take time. And in that time, we've got to learn to live with it and adapt.
That's one of the great things of working in this field is that, I feel like we get to work with some of the most brilliant people as well, because they help to figure out those clever ways to deliver a payload using another virus that isn't something that's infectious to human beings, and those sort of things. It's amazing to know that wonderful scientists out there figure those things out. So I read 2 articles of yours, just to switch gears for a second. The first one looked like it was written in 2016, and it was titled Advice to My Younger Self. My first question for you is actually, if you rewind the clock, so it's been for almost 5 years now since you wrote that article, what advice would you give your younger self now?
Okay, so the advice I'd give my younger self, and thank you for reading my articles, It's an interesting time to look back and reflect, and it's one of those things that I learned fairly at a young age, to self-reflect and think about things. And if I look back on the article now and, what I would say to myself then, what I would say now, the only thing I would change is something I've learned through my career and through my life is be yourself. And I don't just mean that in a flippant way. What I mean is, in every interaction with people, I used to try and adapt quite significantly to their behaviors and their character, and to be something different. And I pretty much failed at it. I tried really hard, and I had so much enthusiasm and passion that I could get by with it, but it didn't really work for me. And so over the last probably 10, almost 20 years, probably 10 years at least, I've just learned to just be more comfortable in my own skin, if that makes sense. Be me and be true to my behaviors, not trying to adapt to a situation as much, and try and just keep your core values and your core behaviors as clear and as open as you can. And the one that I've really engaged with and committed to is honesty. And it really is something I would go back to my younger self and say, look, that is your core behavior. That is you to a T. That's how you're going to be. just be honest, be open, be direct, be blunt. And my old team in my previous role, we did some 360 feedback, and one of the bits I really loved about it was there was a point where one of my team said, Tim can be honest, brutally honest. And I found that quite interesting. And in discussion with the people, that they actually liked it, because if you're brutally honest and if you're honest all the time, it means if you're giving somebody good news or bad news, they know that it comes from an honesty. It doesn't come from a negative perspective. It just comes from honesty. So that's, if I look back on that article, that's where I'd go with it. If I can tell you a little story, that article was something I'd committed to and I really struggled with, and I got writer's block, and I was racking my brains trying to think what I was going to write. And one of my colleagues pulled out of a presentation at a university for graduates. And so I went and gave the presentation. And that presentation was written. I went and gave the presentation. And on the train on the way back, from spending an hour presenting and then an hour talking to these young graduates, in the hour on the train coming back, I actually wrote the article. So it took me less than 45 minutes to write. But it was this inspiration from working with these kids and talking to these graduates that were coming out of university. It just all started to click into place in my head. And it was just, I look back on it today, it's still one of the proudest things I've written. It's such a kind of clear and open and honest discussion, and something that I think everybody can relate to. So yeah, honesty is what I would do if I had to go back and talk to myself.
Yeah, it is interesting. So there's an author by the name of Seth Godin who says that there is no such thing as writer's block. The fact is that you can write. That's not a problem. The fact is it might be crap. It could be really bad writing, and you just might not want to release it whenever it's done. But I read it, and yes, I think just in general, really good advice. I ran for a number of years, and your whole comment about being honest or being true to yourself, one of the things that you learn early on is that you can't actually match somebody's foot for footsteps whenever you run.
Yeah.
And whenever we think that we can match our personality to somebody else, it's the same thing. You can't necessarily be somebody that you're not. You have to be you. And so, yes, that genuine nature and being honest is just a great attribute to you as well. That you found that in the midst of your career as well.
Yeah, just wish I'd found it earlier.
All right. And so the most recent article, right? So it was titled How Bioanalysis Supports the Development of New Cell and Gene Therapies. I guess one of the things that I've extracted from the article is just the real importance of, monoclonal antibodies is really important. They're all over in the news right now in terms of supporting people with COVID, and just the alignment of bioanalysis with the research around this, as well as it seemed there was also an alignment as well with next generation sequencing. And so it just seemed to me like, that the article really expressed or drew those items out.
Yeah, definitely. Cell and gene therapies at this moment are becoming more and more exciting. I've spent the last probably 5 years working on a couple of different spaces for clients that actually makes it quite hard to talk about them, but working with things like CAR T-cell therapies. And when we start investigating what they can do, when you can take somebody who's got a gene that is not functioning correctly and you can cure them by giving them a gene and getting the body to express the proteins themselves is just incredible. Looking at some of the data on the CAR T cells, they're taking late stage cancer subjects who are patients who are their Hail Mary passes, and if we get this right, we can basically cure them. And it's so exciting, and the opportunity to do it, but the challenges around it at the moment are still incredibly complex because these are still really new. Still learning what we're trying to do in this space. And so for us in bioanalysis, we've got so many tools, but there's just trying to find the right answers and the right solutions. And we keep working on it. Monoclonal antibodies have been around for a little while, and we've got quite a lot of really nice therapies in that space. The real exciting stuff in that space is when we start combining monoclonal antibodies with something else. So there's a technique called antibody drug conjugates, ADCs, and they are phenomenal because we use the antibody to target the payload, as it's called, to where we want it to go in the body. And then the payload can be pretty toxic and pretty nasty, but it's targeted to particularly tumors. It's targeted to a tumor area, and then it can actually then cause cell death and remove the tumor. So there's so much investment and engagement in that space that we're going to be curing things that we thought were uncurable. And bioanalysis is, right in the middle of supporting that, and not at the forefront of it. That's a totally different space. But in terms of the support that we have to give, it's makes our life interesting on a daily basis. We'll get some incredible therapies over the next 5 to 10 years, I'm sure.
Yeah, it's interesting because I think it was 10 years ago or so was maybe the first time that I heard the term personalized medicine. And now whenever you look at you can get a diagnostic that essentially, lines right up with a potential therapy. I know I've interviewed before Mark Collins from Holomics, and he says that they're using artificial intelligence to support and drive things around applying the right treatment as well. It's just really interesting to me, and also to hear your area of bioanalysis. The one question I would ask back is how much of bioanalysis today is done in the computer, and how critical is the role of, say, the human being in terms of interpreting the data? Or does a lot of this come directly, out of the computer ready to be applied?
Yeah, artificial intelligence, something that in bioanalysis we haven't really got to. The computers are still just a tool, and we still use the pink fleshy thing on the other end, humans, all the time to do all the data analysis and interpretation. It is changing, and it's changing rapidly. Automation has been the big thing over the last 20 years using robotics. And one of the opportunities I got when I joined Celeron was to go in for the first couple of weeks spending our team in Zurich. And they've done some amazing stuff for automating ligand binding assays. And so you take a sample out of the freezer, and then basically it's a robot from that point on to a result. And the human being is sitting at a computer and gets the data up on their computer. So it's quite an incredible step forward to see that happening, and that automation and level of control of the data as well, because if it's done by a robot, pretty much shouldn't be an error. It should be error free. Now, interpretation of the data, we use a lot of complicated software for statistics and statistical analysis when we talk about pharmacokinetics and pharmacodynamics. But really, at the end of the day, we're still looking at humans to then interpret that into the biologic impact, the treatment impact. So yeah, humans are still really important, but I think the use of artificial intelligence, pattern recognition, these kind of things is going to become more and more to the forefront. Probably another 5 years before it starts to really come in, I think. And seeing people like Google getting involved in medicine and pharmaceutical development is quite exciting. They bring so much innovation and power to what we do from a kind of silicon approach. It'll be interesting to see what they do in this space.
Absolutely. Can you tell us a little bit about what does Celeron do? What do they work on? What is their focus area?
All right. Yeah, absolutely. Number one, we're a contract lab, so we don't develop drugs of our own. What we do is we'll work for anybody who needs support and needs help within the industry. The biggest thing we have is we have phase 1 units. So we have clinics in Phoenix, Lincoln, and in Belfast. And those clinics are designed primarily for healthy volunteer studies. So at the start of a drug development process, when you go into the clinical space, so you've done your animal work, the drug's now been agreed to be safe to be used in humans for trials, at least. It goes normally into healthy volunteer trial. And that's what our primary business is, running those. So we bring people into the clinic, and we give them a dose of the drug, and we then make sure there's no adverse side effects or impact of the drug on the body that's going to cause a problem. So that's our kind of major part of our business. From there, we also have bioanalytical, we also have global, so we run trials in subjects on multi-sites across the world, as well as we do data management as well, and we also do regulatory support, regulatory affairs, and help people do, particularly when you're talking about some of your smaller biotechs and smaller companies, we'll supply the support, regulatory support they need to get their drug to patients. That's where we sit. Bioanalytical, we've got a big team in Lincoln and a big team in Zurich. And most recently, we have a really small team in Phoenix, which is actually part of my department. We set up a team in Phoenix because obviously with the global pandemic and COVID-19 hitting us all, one of the biggest challenges we've faced is being able to run clinical trials in that environment, which has caused us no end of problems. So we actually ended up setting up a very small bioanalytical unit in Phoenix to help us support that.
Okay. That was going to be my next question is, so in terms of, obviously, if you look at places where you can track clinical trials and things like that, I've seen a definite acceleration in clinical trials around COVID-19 drugs. But what about for other drugs? Are all of those studies still continuing without pause or halt at this point anyway? I'm sure there was probably a point where all of us were trying to figure out what to do to support clinical trials, but cancer drug development and other things need to continue on, I would imagine.
Absolutely. And it has been one of the biggest challenges of this year is that for a couple of months this year, our clinics actually had to stop operation while we figured out exactly how to operate in the COVID environment. And every other clinic in the world was in the same environment, in the same situation. So yeah, there has been a definite pause on all clinical research for a while. We're now moving forward. We're finding ways to do it. But again, when you start talking about patient trials, so beyond the phase 1 trials, how can you say to people, we really need you to come into the hospital for a drug trial, for follow-up appointments, for just to get blood drawn, and when it's not a critical situation. So it is causing some major problems with recruitment of people onto trials and continuation of trials. So there's definitely been a slowdown in that space. Certainly an acceleration for COVID, but for everything else, it's been more challenging. It is starting to we're seeing a path forward now, and those trials are starting to open up again, and people are starting to get their research moving in the right direction. But yeah, we're still not at the global research capacity that we were at this time last year because of COVID. So, I mean, if you take the clinics, for instance, just the phase 1 clinics, we're running them socially distant. So within the clinical space, we have less subjects per ward, per room, per space in the clinic. So we have to have less capacity. So it's making scheduling and planning and everything a little bit more challenging. But everybody's doing it. Everybody's trying to find ways around it and make it better and make it more successful every day. But it is causing some real challenges. But the other side of that as well is there's lots of problems with people actually going to their own doctors to get diagnosed. So we're finding that certainly in the UK, there's been a lot of discussion around cancer being the major one, but other disease states as well. We're just not going to be getting early detection anymore because people are just not going to the doctor. It's going to cause us some problems down the line, that's for sure. So COVID's going to have a long-lasting effect, but not just in terms of COVID, in terms of other disease states as well.
Excellent. And I'm glad that you're working through all of that and that you found the path. Obviously, it's important work that's going on. So what are some of the biggest challenges that you're dealing with today at Celeron?
Yeah, no, the biggest is COVID, definitely. I'll talk about some of the other ones as well, but COVID's the biggest one. So when the pandemic hit, and I'm going to steal a bit of thunder from my colleague Sarah Peters, who's presenting a presentation at the European Bioanalytical Forum, the virtual meeting, which is happening this month. Got a really nice story about how we managed to open the clinics. So prior to my time within Celeron, my colleagues had developed a PCR assay, so for detecting viral load within subjects, which was for COVID-19. So we have managed to open the clinics by being able to test people and prove that they are negative. They don't have any viral load within their system. So that's allowed us to put people into a clinical setting, which, if you can imagine, is rooms full of people, which is the last thing you want to do in a pandemic situation, is to go into a room full of other people unless you have to. So that's why we're using social distancing. We're using testing to make sure that everybody going into that environment's clean. So that's been our biggest headache. The biggest challenge we've got right now is, can we get to a point where we do that without social distancing so we can open the capacity back up and run at full speed? So we're working on that really hard at the moment, trying to figure that one out, trying to work out where we talk about in sports, there's all these point of care tests being used, and they're not as specific or selective as the PCR test that we've got and other labs have got. Ours is incredibly sensitive and it's very good. It does a great job for us and can detect really low levels of virus. But when we start comparing it to things like the point of care tests, they're not as sensitive. So it's about trying to find a balance where we can move to using, the problem with the PCR test is it's called the brain tickler. It's a nasal pharyngeal swab. So I don't know if you've had it, but other people will have had that are listening. You have to put the swab almost right up your nose to the back, and then it's quite uncomfortable and painful. We're trying to come up with different ways of doing it. But the point of care test, most of them don't use that. They either use a buccal, which is the inside of the mouth, or they use a normal nasal swab, which is lower down in the nasal cavity and not as uncomfortable. But they're not as sensitive. So the biggest challenge is trying to figure out where all the different testing and assays fit into the being able to open the clinics at full capacity. So that's one thing that, biggest challenge I'm facing right now. The other challenges are really around these cell and gene therapies and new modalities. We're a very heavily regulated industry, obviously, because we're looking at human safety and keeping people from taking drugs that are going to cause them harm. And things like the FDA and the MHRA and the EMA, and so that's the US is the FDA, UK is the MHRA, and the European body is the EMA. Three kind of main ones that I deal with on a daily basis. Their guidances change on a fairly, they don't change, the guidances don't change, but the interpretations can change fairly rapidly and fairly quickly from year to year. So we have to be nimble on that. But when we get into cell and gene therapy, we don't have guidances. We're still such a new area that we're still trying to manage the regulations to the best of our ability. And so those give us a lot of challenges, trying to work out how to apply regulatory requirements which talk about control, reproducibility, accuracy and precision to assays where we just don't have those levels of control. Things like flow cytometry, it's a fairly esoteric way of doing it still. Even when you try and automate the gating of a flow cytometry panel, it's not easy to automate. So again, that's where we're hoping artificial intelligence and things like that will come in the future. But at the moment, it's done by a human being, and I term it's almost artistic, to be sure, the way that they do it. And there's a huge level of understanding of what they're looking at required for them to actually be able to gate the populations to identify which cells they're looking at. So the biggest challenge, for me outside of COVID is really in those regulatory spaces and making sure that the data that we are producing for our clients is going to be accepted by all the different regulatory agencies they're going to submit to.
Very good. Thank you.
Yeah.
We're at the point in the show where I ask the same three questions of every guest. And what I'd like to do is to ask you, what inspires you?
People inspire me. Throughout my career and throughout my life, it's the adaptability and the human spirit inspires me more than anything else. And I try to be a role model to people around me, and I try to learn and take things from everybody I work with. And that in every way, I have 3 children and I do quite a lot of coaching in different sports over my life. And I find that I learn as much as I teach, if that makes sense. So when I'm working with even like an 8-year-old hockey player, or I used to coach fencing, when I'm working with kids who are fencing, you're learning from them about how you can get better at what you do and how you can live. Yeah, human people. And in my career, and when I look back over the last 25 years, the bits I'm proudest of that inspires me is the people that I've worked with that I've helped move their careers forward and how well they've adapted and how well they've learned. And just that side of it really inspires me more than anything. You know, I'm one individual. I have a set of knowledge and experiences. And if we put a group of people together, they can be so much more powerful. And people, definitely, inspiration comes from people.
Absolutely. And it is an interesting thing, though, that as you teach, you seem to learn more.
Yeah, absolutely.
Primarily, and I almost would say the same thing about the podcast for me. It's through hearing and going through these conversations for me, it's always a learning experience. I learn a lot, obviously reading an awful lot about what all of you are doing in the industry, but it really is interesting to me how much being a teacher you learn just by having to answer the questions that people are asking you. And so I think that's spot on.
Yeah.
What concerns you?
Concerns me. Politics.
Yeah, well, good point in time to be concerned about that.
But
as we're recording this, the presidential race is not over in the U.S.
But again, maybe not in a direct way, is particularly in the U.S., the cost of medicines and the development of drugs really is ruled by politics. It's ruled by, governments. And we set limits on drug costs and how much companies can charge for their medicines through different legislation. But at the same time, we keep making the hurdles to get medicines to market higher and higher. And I can understand why we do that. But at the same time, that makes it more expensive. So there's a balancing act here that I don't think we've ever managed to get in a risk approach. Nobody, if you go back into the 1960s with thalidomide, nobody wants another scenario like that. Or T-Genero in the UK. We want to make sure that we're safe. We want to make sure that we're secure. But there's an awful lot of the regulations and the requirements that are set in stone and keep getting expanded, which I'm not sure really add to safety and really add to security of the medicines we're trying to develop. So for me, I think we've got to, because politics is a short-term thing with, governments changing hands in the US every 4 years, and in other countries it can be different numbers of years. It does seem to become almost a political pawn, and it's not really being given the same focus and attention to make sure that we're taking the right risk-based approach. So if you're only in government for 4 years, you're not going to make a risk and change a process which is not going to affect things for 10 years. And I think that particularly with drug development, with the process, it takes a long time, 10, 15 years to get a drug from concept to the market and to the patients that need that drug. We need to make those hurdles lower, but without reducing the safety. And somewhere along that line, we're going to have to take a risk-based approach. And I think that with the way politics is set up, we're not going to get that. It's always going to be the highest possible bar of safety so that they can't be the government that allowed another thalidomide to happen. And so for me, I kind of understand it, but I just don't know how we change it and how we fix that. So that concerns me, is looking forward that it's going to get harder and harder to get a drug to patients. And I have people I know in the FDA, and they work really, really hard to try and make it as easy as possible within the rules and regulations that are set. So it's a difficult one. Politics, for me, in that space really is a problem.
Definitely a different sort of answer than where I thought this would. That I thought that would have gone. But thank you for that, because I do think it's interesting, because as you watch Project Warp Speed, which is the government's accelerator around COVID and treatments and diagnostics and things like that, the one I guess is somewhat of an outsider to that environment. The one thought that I had was, or hope that I would have, is that the overall government would learn, hey, maybe we could do this do something to speed things up all the time, that might be different, but your scenario is probably a lot closer to reality than mine is.
We've got better at it over the last few years. I mean, when I started in the industry 25 years ago, the only area where you could get orphan status, where you could get a drug into people without all the hurdles, was oncology. And we're getting better at those kind of orphan status situations. The FDA have done a really good job on that over the last few years, trying to come up with these accelerated pathways to get things to patients faster, particularly in the really unmet needs. But again, I think we could be doing a lot more from the top down, from the political side, if we actually were able to take risks and be a bit more less conservative about it. So.
Very good. And what excites you?
What excites me? Probably the new developments, the new modalities, the cell and gene therapies. The fact that I think in 10 years from now we'll be treating things that are just considered as untreatable. That really excites me. The fact that we can take a person's own immunological response and engineer it to solve problems that we just couldn't do before. So the CAR T-cell therapies have really excited me and blown me away over the last few years that I've been watching those, and watching the first one get to market has been incredible. Really excited by that. That really, excites me. The future is bright in terms of medicines, even though we've got the hurdles and the politics and everything else I've talked about. Companies are innovating and they're driving the processes forward to get patients treatments faster and quicker and as to the best of our ability. So that excites me. And part of that is also the challenges for Bioanalytical, because it's getting more and more I'd like to use the word difficult in that space, but I don't mean difficult. I don't mean it in a negative way. The challenges are getting more opportunity for the bioanalytical scientists to flex their muscles and flex their brains. And if you go back 25 years ago, bioanalysis was seen as a side process, and we had to get some exposure data. Now we're seen as part of the decision making. We're now seen as part of the processes of trying to understand. And you talked earlier about patient specific medicine. So we're doing personalized medicine, and that again is such a great thing, that we can phenotype and genotype a subject and identify which treatment will work for them. And bioanalysis has driven that. We've driven that through, and you mentioned next gen sequencing. That's going to be one of those big things that really drives that forward. So I'm excited around the new modalities, the cell and gene therapies, potentially some of these ADCs. I've been working on delivery systems since 1999, 2000, looking at lipid nanoparticles, and those are now becoming such a key part of the industry to deliver oligonucleotides into the body, putting them into nanostructures to protect them, to give them some kind of stability, to get them where we need them to go. So really excited around those kind of things.
Excellent. And so, Timothy Sangster, I wanted to thank you for being on the show. Thank you for spending time with me and for providing all your insights.
It's been my pleasure. Thanks, Don. Have a great day.
Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it. And last but not least, rate the podcast. Thank you again.